Good day, grade 12 learners, and welcome to this video where we will be looking at the location, structure, and functions of the DNA molecule. You would have remembered that in the previous video, we just briefly introduced DNA, the code of life. So in the previous video, based on your exam guidelines, we looked at revision of the structure of a cell. We looked at two types of nucleic acids, namely DNA and RNA. And then we also said that nucleic acids consisted of nucleotides. In this video, we will be looking at the location, structure, and functions of the DNA molecule. And the first aspect that we'll touch on will be the location of DNA. So firstly, we will look at DNA being present inside the nucleus, inside the mitochondria, and inside the chloroplast if it's going to be a plant cell. So if you have a look at this diagram over here that clearly shows the location of DNA molecule inside a cell, you will see that DNA, which is deoxyribonucleic acid, is mostly located inside the nucleus of a cell. And this was something that we would have touched on in the previous video as well. So most of our DNA, most of our deoxyribonucleic acid is going to be found inside the nucleus of the cell. And remember in the previous video we said that in the nucleus we are going to have the chromatin network. Now that chromatin network is eventually going to condense to form chromosomes. So inside this nucleus here you can clearly see that there are chromosomes, and these chromosomes have formed from the chromatin network. So therefore it follows that on these chromosomes we are going to have our DNA. So on these chromosomes you will have the DNA. If you have a look here, you will see that the DNA double helix structure, if you move right down to the bottom of this diagram, the DNA double helix structure is wrapped around certain proteins referred to as histones. And that DNA double helix is wrapped around proteins, and all of this is actually found on the chromosome. And this chromosome originated from the nucleus. It's lying inside the nucleus, and it formed from the chromatin network. So let's just go back. We say that DNA is located inside the nucleus. The majority of our DNA located inside the nucleus. The nucleus contained the chromatin network, which condensed to form the chromosomes. So therefore, on each and every chromosome, we are going to have our DNA double helix structure that is generally wrapped around certain proteins which we refer to as histones. If you have a look further down at the bottom, you will notice that the DNA is basically the DNA molecule is forming a double helix structure, and that is basically going to be the structure of the DNA molecule, which we'll look at later on. So if we're looking at the basic location of DNA, we see that it's located inside the nucleus. So the nucleus comes out of a cell. DNA is also located inside the mitochondria. So if you look at this general structure of the cell, we have a mitochondria there. DNA is also located inside the mitochondria. And if we are looking at plant cells only, they will also have DNA located inside the chloroplast. So just to go back to what we mentioned earlier on, we see that on the chromosome, we are going to have our DNA. And here you can see you have the DNA double helix structure. And if you are looking at one short segment of DNA, if I have to take one short segment of DNA that's found on this chromosome, that's going to code for a specific characteristic, then that is referred to as a gene. I'm going to repeat that. If I take one short segment of DNA found on the chromosome that's going to code for a specific characteristic, then that is referred to as a gene. And there are going to be a number of genes found on the chromosome. Why am I saying that? Because they are DNA. DNA is located on the chromosome, and it's located all over the actual chromosome. So just going back to our exam guidelines, so far we've touched on the location of our DNA strand. We said it's present inside the nucleus. That's when we refer to it as nuclear DNA. It's present inside the mitochondria when we refer to it as mitochondrial DNA. And then lastly, it's also present inside the chloroplast, but that will only be inside plant cells. We will now move over to the next important aspect, where we will look at a brief history and discovery of the DNA molecule. We will focus on certain scientists such as Watson and Crick, Franklin, and Wilkins. So if we're looking at a brief history of the discovery of the DNA molecule, firstly in 1952, we had Rosalind Franklin and her assistant Morris Wilkins, who researched the structure of DNA using X-ray diffraction images. So the most important aspect that you need to take note of here is that in 1952, Rosalind Franklin, she came up with a certain type of X-ray that helped with the work to come up with the structure of the actual DNA molecule. Secondly, James Watson and Francis Crick did independent research on DNA. What do we mean by this? James Watson and Francis Crick did their own research to try to come up with the actual structure of the DNA molecule. Watson and Crick then proposed a 3D double helix model for DNA in 1953. So that actual structure of DNA that we know it to be today, it was proposed by two scientists, namely James Watson and Francis Crick, as they came up with the structure of the DNA molecule, which is a double helix structure. And this double helix is a 3D structure. In 1962, Watson and Crick received the Nobel Prize for the discovery of the structure of the DNA. And Wilkins, that is Morris Wilkins, received an award for his X-ray photography. You will notice that James Watson, Francis Crick, and Morris Wilkins received awards. However, Rosalind Franklin, she also contributed to her X-ray that she provided, but she was not awarded for anything. Therefore, she did not receive any recognition for her works. So it's just a photograph of James Watson and Francis Crick in their work trying to come up with the structure of the DNA molecule. And as we mentioned earlier on, that structure that they proposed was a DNA double helix structure, which has been coming in the form of a 3D model. Earlier on, we also mentioned that Rosalind Franklin came up with an X-ray. So this is Rosalind Franklin, and this is what the X-ray looked like, which she proposed to help to come up with the structure of the DNA molecule. And the X-ray that she set forward, the name of the X-ray was referred to as Photo 51. The rest of these are simply pictures of the scientists mentioned, namely James Watson, Francis Crick, and Morris Wilkins. So going back to our exam guidelines, thus far we've touched on the location of DNA, the brief history and discovery of the DNA molecule. We focused on scientists James Watson, Francis Crick, Rosalind Franklin, as well as Morris Wilkins. We will now move on to the structure of the DNA molecule. We will look at the natural shape of the DNA molecule being the double helix. We'll look at how each strand of the helix is made up of a sequence of DNA nucleotides. So if we look at the DNA structure, as mentioned earlier on, we see that DNA consists of two molecules that are arranged into a ladder-like shaped structure called a double helix. So that double helix structure, which I mentioned earlier on, it's going to comprise of two molecules that are arranged in a ladder-like structure called the double helix. A molecule of DNA is made up of millions of tiny subunits, and these subunits are referred to as nucleotides. Now, you would have remembered in the previous video we said that the building blocks or the monomers of DNA are referred to as nucleotides. Now this is that exact same nucleotides that we mentioned in the previous video. So the actual molecule of DNA, it's made up of millions of subunits and they are known as nucleotides. Just to recap what we mentioned in the previous video, we said that each nucleotide, it comprises of a phosphate group, a pentose sugar, as well as a nitrogenous base. So if we have a look at this diagram, it basically shows us a picture of what the DNA double helix actually looks like. So we mentioned that the DNA structure is such that it comprises of two strands, a double-stranded molecule that then becomes coiled up and it then forms this three-dimensional structure as you can see over here. If we looked at the nucleotide, remember we said the nucleotide comprised of the phosphate group, a pentose sugar, as well as a nitrogenous base. Now, if you are looking at our nucleotides, we said it comprises of the phosphate group, the sugar, and the nitrogenous base. Now, specifically because we are looking at a DNA molecule, you need to remember the following. The phosphate group, the type of sugar that we are looking at here will be deoxyribose sugar. So the type of sugar in the DNA molecule is referred to as deoxyribose sugar. So you'll have deoxyribose sugar here, deoxyribose sugar there, deoxyribose sugar, and deoxyribose sugar. When we are looking at the types of nitrogenous bases in a DNA molecule, we'll have one of four types of nitrogenous bases. It's either adenine, thymine, guanine, or cytosine. So I'll go back to that. In the DNA nucleotide, the DNA nucleotide comprises of a phosphate group. The type of sugar will be deoxyribose sugar. There are one of four possible types of nitrogenous bases. That is adenine, thymine, guanine, and cytosine. So each base will generally bond with one other specific base. So in your nucleotides, if we're focusing on our nitrogenous bases, if I just have to go back, these are the nitrogenous bases, adenine, thymine, guanine, and cytosine. We're saying that some of these nitrogenous bases are going to form pairs with each other. And generally, adenine will only pair with thymine and guanine will only pair with cytosine. What does this mean? This means that adenine and thymine are going to form base pairs and cytosine and guanine are going to form base pairs. Therefore, we can also refer to these nitrogenous bases as being complementary base pairs. Adenine and thymine and guanine as well as cytosine. Just a reminder that these nitrogenous bases, adenine, thymine, guanine, and cytosine, are found on the nucleotides, and they are the possible nitrogenous bases that you will find on a DNA molecule. So if we have to look at the general structure of a DNA molecule, and if we have to open up the structure of a DNA molecule such that we're looking at a two-dimensional structure, then this is basically what you will see. So if you have to look at the majority of the questions that you should be getting this year, either in your control tests or in your June exams, trial exams, or NSC final exams, if they generally test you on the structure of the DNA molecule, majority of the questions will have the DNA molecule opened up like this, very similar to this. It will be opened up and you will then be asked to examine the structure of the DNA and they'll ask you questions respectively. So I'm just going to go through everything that is possibly stated on this diagram so that we know everything about the structure of the DNA molecule that we possibly need to know. So firstly, you will notice that it's a double-stranded molecule. There's a strand on the right, there's a strand on the left. Secondly, each Each strand is going to comprise of a number of nucleotides. So you have one nucleotide there, 2, 3, and 4. So there are 4 nucleotides on the side. There are 1, 2, 3, and 4 nucleotides on that side. So 4 nucleotides on the right, 4 nucleotides on the left. In total, there are 8 nucleotides on this particular structure of DNA. You will notice that they have labeled the nitrogenous base. So this is a nitrogenous base, the nitrogenous base, and they are color coded. Each nitrogenous base, very, very important, each nitrogenous base is joined to the complementary base pair. I'm going to say it again. Each nitrogenous base is joined to the complementary base pair. So adenine will join to thymine, guanine joins to cytosine, and they are going to be joined using a weak hydrogen bond. So at the bottom, here's the label, weak hydrogen bond. This weak hydrogen bond is going to ensure that all the nucleotides on this DNA strand is going to be joined. And because all these, all these nitrogenous bases are going to be joined using a weak hydrogen bond, and because all these nitrogenous bases are joined using a weak hydrogen bond, it then allowed the DNA structure to be such that it's going to have a double-stranded structure. So 2 strands have now been joined together since the complementary base pairs of the nitrogenous bases have been joined using a weak hydrogen bond. Now, another important factor to remember is that some of these nitrogenous bases are going to be long and others are going to be short. And what exactly do I mean by that? If we have a look at adenine and guanine, you'll notice that the adenine is longer and guanine is longer compared to thymine and cytosine, because you can see cytosine and thymine are smaller nitrogenous bases. So since adenine and guanine are longer, they are generally going to be double-ringed. They are referred to as our purines, and this is a term that you need to remember. So adenine and guanine, they are double-ringed, they are generally longer, they are referred to as purines. On the other hand, thymine and cytosine, they are smaller, they are single-ringed, and therefore they are referred to as pyrimidines. So please remember this terminology. Your pyrimidines is going to be thymine and cytosine, they are going to be single-ringed, and purines are going to be adenine and guanine, they are going to be your double-ringed, they are basically longer. Then all these black circles that you have on the ends are referred to as your phosphate group. You would have already known that. And the type of sugars that we are looking at in each nucleotide is going to be deoxyribose sugar, since we are looking at the structure of a DNA molecule. Just one last thing to touch on. If you look at the phosphate and the deoxyribose sugar, they are also joined with a certain type of bond, and that bond is referred to as a strong sugar phosphate bond. This bond is a strong bond, the strong sugar phosphate bond, and remember that the weak hydrogen bond that joins the complementary nitrogenous base pairs, that is a weak hydrogen bond. And we'll learn later on as to why this bond needs to be a weaker bond, because possibly it's going to need to break. So if you have a look at this diagram here, it's just an additional diagram that states very similar to what we looked at in the previous one. At the bottom, here's an important note that tells you how to recognize a DNA molecule, and this is a skill that you need to grasp very quickly, because it is a question that could possibly be asked in an exam. So how do we recognize a DNA molecule? Firstly, it's double-stranded. Secondly, it contains nitrogenous bases thymine instead of uracil. Uracil would be the nitrogenous base that's found in an RNA molecule. We'll learn about that later on. So it contains the nitrogenous base thymine. Then adenine always joins with thymine and guanine always joins with cytosine. So since they are joined, the DNA molecule is going to form a double helix structure. It's going to have 2 strands. since the complementary base pairs have now been joined using their weak hydrogen bonds. If we have a look at the functions of our DNA, there are two basic functions that you need to be aware of. The first one being sections of DNA forming genes carry hereditary information. So you'd have remembered earlier on we mentioned that our DNA is located on our chromosome. And you have short segments of DNA found on the chromosome, and that short sections of DNA are referred to as genes. Now, genes generally carry hereditary information. And what is hereditary information? This information is generally inherited from our parents. So certain characteristics like your eye color, your skin color, your hair color, the hair texture, these are things that you inherit from your parents, and they are referred to as hereditary information. So the first function of DNA, it's going to provide hereditary information in the form of genes, and like we have said, the genes are going to be found on the chromosome. Second important function of DNA is that DNA contains coded information for protein synthesis. So what exactly do we mean by this? Later on in this topic, towards the end of this topic, when we end off DNA, the code of life, the last aspect of this topic, we will touch on protein synthesis. We will go through the process that is going to allow us to see how a protein is actually formed. So DNA is going to contain the coded information for protein synthesis. This simply means that DNA is going to provide the code. It's going to provide the first message in order for protein synthesis to actually occur. And what does protein synthesis mean? The process where we are going to be making that particular protein. So those are the two important functions of DNA. So in summary, this video discussed location, structure, and functions of DNA. We looked at the location present in the nucleus, mitochondria, chloroplasts, and plants. We looked at the brief history and discovery of the DNA molecule, the structure of the DNA molecule. The natural shape is a double helix. Each strand is made up of a sequence of DNA nucleotides. We said that there are three components in the DNA nucleotide. The nitrogenous bases were linked by weak hydrogen bonds. 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. The pairing of the bases in the DNA occurred such that adenine paired with thymine, guanine paired with cytosine. The type of sugar found in a DNA nucleotide is deoxyribose sugar, and then lastly, the nucleotide also comprises of the phosphate portion. We also looked at a stick diagram of a DNA molecule to illustrate its structure, and then lastly, we looked at two important functions of DNA. DNA makes up the genes which carry hereditary information, and secondly, it provides the coded information for protein synthesis. Thank you very much for taking your time to watch this video. In the next video, we will be looking at the process of DNA replication as well as DNA profiling.